This lecture will cover the other gram-negative bacteria in the lab tests that have not been previously covered in other lectures. With your Neisseria and your Moraxella species, these organisms, if they are suspected, may be inoculated on the sheep blood agar as well as chocolate agars. And these cultures will be incubated 37 degrees with 3 to 5% carbon dioxide environment, such as regular atmospheric environment. And then to actually culture this organism, you would need to do it right away just to make sure you maintain the viability of it. Now, for culture with the gonorrhea, Neisseria gonorrhea, these organisms should be collected on a specific swab, so it would be a dacron or a rayon fiber swab. And I will tell you, if you send the wrong swab to the lab, they may reject it. So you need to make sure that you are collecting the right swab for the source, and because if not, then it can actually be an environment for that organism in which it cannot grow properly. So, or it doesn't maintain that organism in transport. So, making sure that you have the proper culture swab is important. And so these organisms for the Neisseria gonorrhea should be cultured on the chocolate agar, but it also can be placed on some of these other agars, which would be like the Thermartin or the modified Thermartin. And the reason why these other agars would be used, because you can see an image here where you have a chocolate medium. So in particular, if you were doing a vaginal swab looking for to try to diagnose a patient with gonorrhea, then the normal flora that would reside within the vaginal area would be abundant. So if you were to plate it on culture it onto a chocolate agar, then you would get a lot of growth for the other organisms that would be present. And so if you use something that's specific for like a vaginal culture or a genital culture, where some of the antibiotics in there would prevent growth of some of that normal flora, then it helps cut down the organisms that you're looking at. So you can see in comparison here, where at the bottom you have, they actually did this with a rectal swab, where you have a lot of normal flora that has grown on that chocolate agar, versus the modified Thermartin media, where just the Neisseria has grown. So some of the different test methods for Neisseria and Moraxella would include nucleic acid-based testing. And so with these tests, the instrumentation nowadays with the automation will actually look for multiple organisms off of one swab, one sample. And so if you were doing like a GC probe, the gonorrhea and chlamydia probe, then you're looking for both of those organisms off that one sample. Your identification of your organism and how you work it up is going to depend a lot on the source. So it's very important that the sources are labeled properly and that it includes where the source site is. So for example, a rectal source or a vaginal source. Any isolate from a child or anyone suspected of sexual abuse, it definitely has to be identified. It definitely has to be worked up. Isolates from sterile body fluids are completely identified. Isolates from genital sites can have a presumptive identification just based off of the spot test. It's oxidase positive gram-negative diplococci that grew on the select media, so we can presume it to be the gonorrhea. Eye and ear cultures can yield oxidase positive, gram-negative diplococci, and hydrolyzed Trubitin would be considered a presumptive identification for the Moraxella catarrhalis. The carbohydrate utilization test is used to help identify mostly your Neisseria species. And what this test does is uses the different carbohydrates, glucose or dextrose, as it's sometimes referred to, maltose, lactose, and sucrose. And these sugars will all have, or carbohydrates will all have a phenol red pH indicator in it. So if it is, the organism is able to utilize it, then it will change the pH, and then you would get that yellow color would be demonstrated within that particular tube. So I think in one of the previous lectures we talked about testing these carbohydrates on a tube test. And so here you've got an example of a little bit more of a modified test where you have each of these little whales that have a one of these carbohydrates in it. So you can see here C is control, and so you've got each of these little whales. And then here you have your glucose. It turned yellow, so this would be considered positive. Your maltose is yellow, positive. Lactose is positive, and then your sucrose is negative. You also have a DNA test as well as the beta-lactamase test. But for this particular organism, because it utilizes all the way through the lactose, then it would be the Neisseria lactamicea. So with this, you would also want to avoid putting these into a CO2 environment, because that can affect how these organisms utilize these carbohydrates. So here we have a chart to help compare these reactions in these organisms. There are several species of Neisseria that can also be identified using this test method, but we've not covered those, and they're not always clinically relevant, so we're just covering the ones that you'll probably experience more in your career. And so an easy way to remember these is your glucose is gonorrhea, your maltose is meningitis, and then your Moraxella does not use any of these. So you can see just glucose for gonorrhea, glucose and maltose for meningitis, and then your catarrhalis. Cats don't like sugar. I don't know, do they? So cats are negative all the way through. Campylobacter jejuni would need to be cultured on selective media, so your Campy media. You can have Campy blood agar, or you can have other Campy medias. And so if you were doing a stool culture, the technician is going to set up a couple of different selective agars, and that's because they're going to culture one at 42 degrees Celsius and one at 37 degrees Celsius, just to make sure they differentiate it from another organism that could grow and appear to be similar, that wouldn't be considered pathogenic. With your blood agar, it will grow on a blood agar plate, but it can take a little bit of time to grow.